replication. Let's continue that. And you should recognize this feature from the animation video that we watched twice on Monday. That is when the double helix, to initiate replication, the double helix is unzipped, forming a replication bubble, and each bubble has 2 replication forks. And again, because of the anti-parallel nature of DNA, that the new strands are produced in opposite directions. So we have, again, the leading strand synthesis directed into the replication fork, and the lagging strand away from the fork, and has to be repeatedly primed, okay, producing the Okazaki fragments. Okay, and it's interesting that the replication origin is rich in a certain base pair. Anybody want to guess what that base pair is? It's rich in, now there are 2 types of base pairs. There's the AT base pair that's held together by 2 hydrogen bonds. Then there's the CG base pair. We're talking about the 2 nucleotides that hydrogen bond between the 2 complementary strands, and hydrogen bonds with 3 bonds with CG. So if you've got to unzip the molecule, which to make it easier for the cell, which base pairs do you think are more common here? Well, if you're unzipping something, what's going to take less energy to unzip it? Something that's held together with 3 bonds or versus 2? 2. So this is rich in AT base pairs, the replication origin. That's one of the ways that scientists kind of look for these things when they have a genome sequence. Okay. So it's rich in AT base pairs. Now we look at some. Essentially, replication is mostly the same in prokaryotes and eukaryotes. Of course, eukaryotes are descendant of prokaryotes. But there are some important differences here. If we look at the rate of replication between prokaryotes versus eukaryotes, for prokaryotes, they can replicate DNA at a rate of about 1,000 base pairs a second, making a new complementary new strand. Okay? Which means that for E. coli, it has a genome size of about 4 million base pairs, approximately. It takes about, with a single origin of replication, we represent that, we always abbreviate that in molecular biology as ORI. That stands for, just right out the first time, origin of replication. Oops, we spell replication, start with an R. They have a single origin. At this rate, they can copy the genome in about 40 minutes. With one single origin replication. Okay. We have a different scenario for a eukaryote. Okay. In eukaryotes, and I'll focus on us, we're a eukaryote. But typical for a mammal like us, we have about 3.2 billion base pairs, so we have 1,000 times more DNA. Okay? And the rate of replication for us is only about 50 base pairs a second. Ooh, it's slow. Why do you think it's so slow? What could possibly slow it down? It's like 20 times slower. At this rate, to replicate just one chromosome, with a single origin replication, let's say this chromosome, and typically a human chromosome is around 150 million nucleotides long, base pairs in size. It would take around 800 hours. Ooh, that's too long. So in eukaryotes, there are multiple replication bubbles, multiple origins. In fact, on a chromosome, anywhere between 20 and 80. And they're spaced about 300,000 base pairs apart. Which means if we have 20 to 80 origins, we can complete in about 8 hours. Versus 800. I don't know how many days 800 hours is, but divide that by 20. Let me divide it by 24. I'm just curious here. About 33 days, that's about a month. Take about a month to replicate a single chromosome with a single origin. So as I said, we have anywhere between, I gotta look at my notes, I already forgot, 20 and 80 origins. And they're spaced about 300,000 base pairs apart. You get it done in about 8 hours, okay, with 20 to 80 origins. They're typically about 300,000 base pairs apart. And at this rate, even though it's slow, we can get it done in about, complete the genome, about 8 hours. That's about how long that S phase of interphase lasts. And then these things just fuse into each other. Okay. Here's a photograph of this. This is time 1, time 2, time 3. So you can see the origins, bubbles starting to happen, the replication bubbles at the origins. As they're replicating the new DNAs in red, they just start to spread and spread. Here's an actual photograph here. DNA being replicated. Just my back and do an electron micrograph. As I said, remember the bacterial chromosome is a double-stranded single circular chromosome. Has one origin replication, and again, it takes about 40 minutes to complete that, and then divide into two. Okay. With a single origin. Okay, that's pretty simple, straightforward stuff. Okay, now there's a complication for the linear structure of eukaryotic chromosomes. I'm going to back up the slide just for a second here. In the bacterial case, they have one circle. Replication, the replicates end up, they seal themselves together, complete replication, and then it just pops in two. And each of these ends up in a different daughter cell after binary fission. But for eukaryotes, remember our chromosomes are linear strands, or ends, which means we can't replicate the end of the chromosome. Every replication cycle, the chromosome gets shorter. So let's look at one replication fork here. Okay, you can see here's the leading strand being continuously produced by DNA polymerase 3 toward the replication fork. Here's the lagging strand being reprimed every time. Now remember, the DNA polymerase can only extend in the 5 to 3' direction. It cannot synthesize in the reverse direction, right? So when we reach the end, when this replication is completed here, okay, well, remember the primers, which are made out of what? RNA, they have to be removed and replaced. That's by the action of DNA polymerase 1. Remember, it can chew this off. Okay, all right, but it can't seal the end. Okay, it can't go to the 5 to 3 direction. So every cycle, this shortens a little bit. Okay. And the consequence is that at some point your DNA can't replicate anymore; it becomes senescent. So it really, we call that replicative cell senescence. It really limits the number of cell divisions a cell can undergo, which is kind of a safeguard against cells dividing out of control when the machinery is working correctly. But if your cells of your gonads, testes or ovaries, are mature and gone through those 60 cycles and start to produce gametes for a new generation, you like the chromosomes in your offspring to have a full set, full-length chromosome. They need a way to restore this end. Does this make sense? Right? right? Because it's the 5' end, and synthesis can't go in the it can't go in that direction, right? Okay. So instead, to deal with this end replication problem, there's a special enzyme we have called telomerase. Okay? And it turns out that the end of the chromosomes, they have the DNA sequence at the end of the chromosomes is a special sequence, a repetitive sequence. Okay? So at the end of eukaryotic chromosomes, there are repetitive sequences. That means a sequence that repeats itself. The sequences are called telomeres. And in humans, that sequence is TTAGGG. And it repeats itself over and over. There's the repeat sequence. It repeats itself for anywhere from 2 to 30,000 base pairs. KB, you know what KB stands for? Think metric.
1,000 base pairs.
And kilo base pairs. So KB means 1,000 bases. So we divide 30,000 by 6 and calculate how many repeats are at the end. I like to think of it as like the wrappings around a shoelace. They keep it from fraying. Okay? And that's what these represent here, these little blocks here. These are the telomere repeats. Okay? Now, they have a couple of functions. They're often associated with proteins. We won't go into that. But they help a cell recognize that the end of a chromosome it's really its end and not a broken piece. Because if they're broken, they'll be repaired. Okay? And it also helps to reestablish the chromosome size after replication. And the enzyme that can restore this